Dear students, until now you have learned about that type of anaerobic glycolysis which gives rise to the formation of lactate or lactic acid. But there is another very important part of anaerobic glycolysis or fermentation which gives rise to the formation of alcohol. Usually this type of glycolysis, or rather anaerobic glycolysis, take place in yeast. In this, the glucose is metabolized to pyruvate, of course, as is the case in all the glycolysis processes, but then that pyruvate is converted into ethanol. How it happens? So, here you can see a pyruvate molecule is converted into acetaldehyde. There are, two section of this conversion of pyruvate into the ethanol. In the first section, in the first part of the reaction, the pyruvate is converted into acetaldehyde, and because this is a decarboxylation reaction, carbon dioxide molecules are released from this reaction. That is why the 3 molecule pyruvate is converted into 2 molecule acetaldehyde. It also uses thymine, pyrophosphate, and magnesium ions. And of course, the enzyme is pyruvate decarboxylase because it is giving rise to the decarboxylation of the pyruvate molecule. In the second step, what happens? That acetaldehyde is converted into ethanol, again a 2 carbon atom molecule. Here you can see that NADH is being used, and as a result of this you get the ethanol. From where this NADH2 comes? If you remember this reaction which gives rise to the formation of 1,3-biphosphoglycerate, from glyceraldehyde 3-phosphate you get NADH. Now keep in mind that this NADH now doesn't go in the what we say, this doesn't go in the ETC, doesn't become a part of ETC, is not utilized by ETC because there is no ETC. This reaction is not taking place in mitochondria, it's taking place without oxygen. The overall equation of the reaction is that you have glucose and ADP and inorganic phosphates as the reactants, and in the products you get ethanol, carbon dioxide, 2 molecule of ATP, and water. Of course, you get the 2 molecule of ATP because at the end of the day, it's an anaerobic reaction. The yield in terms of the number of ATP is much less. Also, the hydrogen to carbon ratio remains the same, it doesn't change. The glucose is fermented into 2 ethanol molecules. So 2 ethanol molecules means 4 carbon atoms being utilized and 2 CO2 molecules. So 6 carbon atoms are retained. There is no net change in the number of carbon atoms or number of carbon to hydrogen atoms or ratio of carbon to hydrogen atom in this process. Pyruvate decarboxylase is present in different organisms, but in simple organisms rather. It's present in yeast, we have already discussed. It's present in certain plants, some plants. This use of decarboxylation process in yeast, it is also useful for commercial uses. For instance, for the formation of yeast, it gives, because of the carbon dioxide which is produced, it gives a certain texture and taste to the product. As I have already told you, that because the carbon dioxide is released in the formation of ethanol through yeast in this anaerobic glycolysis, so because of the carbonation, a specific usually swelling of the material takes place. If you are going to make a dough for pizza or whatsoever, it will swell because the carbonation process, carbon dioxide released, and you get the required texture and of course taste also. So there are many commercial uses also. You will learn about it in little bit detail later, and also in much more detail in a specific course which is associated with the biotechnology and use of biotechnology, use of microorganisms in biotechnology. Interestingly, this all important enzyme which leads to the decarboxylation is not present in us, in vertebrates, in all the higher organisms. It's not present, so we can't really convert pyruvate into ethanol. In our body, of course, pyruvate is converted into ethanol, but through the agency of certain yeast or other microorganisms.